Okay, what we're going to talk about today is the process of meiosis. And if you remember from yesterday, our definition of meiosis is when we want to reduce the number of chromosomes, right, in a cell by half, producing 4 1N or 4 haploid cells. And as we talked yesterday and a little more today, our normal body cells go through mitosis, right, but sexual reproductive cells go through the process of meiosis, again, because the goal is to reduce the number of chromosomes in the cell to half. Because if we didn't, then if the cell just went through mitosis, and it would have a full number of chromosomes, or the new cells would have a full number of chromosomes, and if those cells were used to reproduce, then the new fertilized cell would have double. All right, and we said if that was a human, it would have 92 chromosomes, and that would not be good. So what actually happens is, it's basically reproductive cells, right, go through mitosis twice. And we'll take a look at the stages right now. Right. So the first stage, and actually the first series of steps, the first 4 is called meiosis 1. And we start with prophase 1, just like mitosis. And you can see that our DNA, or remember that our DNA has already been copied, right, during interphase. And so it is condensed so we can see the individual chromosomes. The nuclear membrane has disappeared. And in our cell, in our example here, we're going to use 6 matching pairs of chromatids, or 6 full chromosomes. You see that I have 2 green, 2 blue, and 2 yellow. I'd like you to use 3 colors, just like I have, and have 2 of each color, so that you have 6. And then remember that in a human at this point, we would have 92 chromosomes. Right. Moving on to the second stage would be metaphase 1, and in metaphase our chromosomes, right, there's actually a matching chromosome, right, and so what they do is they pair up next to each other right down the middle of the cell, just like in regular metaphase, but instead of a single line, there's like a double line. And so you see, so the 2 green chromosomes line up next to each other, the 2 yellow ones, the 2 blue ones. So do the same thing in your diagram where you have matching chromosomes, right, lining up next to each other, running down the middle of the cell. And again, you can see that our spindle fibers have been released from the centrioles, and we are getting ready to pull the chromosomes apart. And so in stage 3 we have Anaphase 1, and actually I just misspoke. The chromosomes actually don't separate and get pulled apart. Each individual chromosome of the matching pair gets pulled to the side. Right, so the full chromosome on the left, the full green one on the left, and the full green one on the right get separated from each other. Alright, so you see that the chromosomes have been pulled to opposite sides. We still have a full number in the cell, and again, for our example, we have 6. And in a human, it would still be 92. Right. And then our fourth and final stage of meiosis 1, the first series, we have telophase and cytokinesis. And you remember that during telophase, the cell has pinched and was ready to divide, and in cytokinesis is actually when the cells do divide. So we have 2 new daughter cells, and they each now have 3 full chromosomes, because if you remember in the last slide we had 3 chromosomes pulled to each side of the cell. So now that the cell is split, each new cell has 3 full chromosomes. So so in our example we have 1 green, 1 yellow, 1 blue, and we see that our nuclear membrane has reappeared, and these cells are called diploid, or 2N cells. And remember that the variable n represents a number, and in this case it would be the number of chromosomes. So in a human it would be 23, so 23 times 2 again would be 46. And this would be a normal series of mitosis, and it would stop, and the cell would then go back into interphase and do its thing. But with reproductive cells, they go through the whole process one more time. So now we move on to meiosis 2. Alright, and we start right up again with prophase 2. Alright, our nuclear membrane disappears again. Alright, and the individual chromosomes, alright, are getting ready to move and to line up. All right, but what we want to talk about is each individual chromosome now, and this kind of goes back to original mitosis, is made of one chromatid. And you remember that I just drew that at the bottom, and we have our centromere that holds them together. All right, so the cells each have 3 chromosomes in our example, 46 in a human, and they are getting ready to line up and separate. Now we have Metaphase II, and so our chromosomes line up in the middle of each cell. Our spindle fibers have been released, and they are getting ready to pull the individual chromosomes apart. Again, we still have 3 chromosomes in each cell, 46 in a human. And we move on to Anaphase II. And so now the spindle fibers the centromere has dissolved, the spindle fibers have been released, and they've pulled those individual chromosomes, all right, apart. And so the chromatid, now we have individual chromatids, or homologs, which was mentioned in the last chapter, the earlier chapter. And so they're pulled to opposite sides of the cell, and we still have 3 chromosomes, even though they've been separated, in each cell, and that would be 46 in a human. Finally, we have Telophase II and cytokinesis. And so now our cells have pinched and divided, and we are now left with 4 cells. All right, and each of these cells has one individual chromatid, right, or one strand of DNA. So they have half the number of the original cell. So in our case, it looks like they have 3, but remember, that's just 1/2 of that chromosome. All right. So in a human, we would have 23, and then those strands of DNA would relax and loosen and turn into just that pile of loose string, and they would kind of wait and go through interphase until eventually they would meet up with the opposite reproductive cell, and they would be fertilized to form another organism.